Hey everyone, I'm Catherine Tan. I'm a PhD candidate from the Fred Chang Lab at UCSF. So why does cell size matter? Something that I've come to appreciate about cell size generally is that there's all kinds of different kinds of cells in the world. But if you look within each cell type, cells exhibit a relatively narrow range of size, suggesting that size has a physiological range. And that when you look at a growing cell, this change of size is accompanied by a complex dance of coordinating its rates of growth and division, and the rate at which it makes macromolecules, so all the stuff inside. And so I'll be focusing more on this last point here. So as Chris from the Scothine Lab has mentioned earlier, cells do indeed scale their components proportional to their size. And this has been observed in organisms from E. coli to yeast. And this range is considered to be within the physiological size range. However, what's been more recently described and appreciated is that these components may only scale up to a certain limit. And so in studies where you grow a cell overly large, you not only lose this scaling of biosynthesis, but also these macromolecular changes are accompanied by phenotypes such as senescence or the reduced ability to proliferate. And so why is this happening? So this loss of biosynthetic scaling has been attributed to the idea that DNA is limiting. So if you don't have enough template, you can't make all the stuff or macromolecules that are required in this larger cell size. And so it might be important to keep in mind this ratio of DNA to your cytoplasm. And that's why we're so interested in biosynthetic scaling, is because we believe the changes in the macromolecular content of the cell are related to its intracellular biophysical properties. And so a study from the Amon lab a couple years ago looked at overly large mammalian cells and observed an overall decrease in macromolecular concentration and an accompanying cytoplasmic dilution. And so this overall suggests that a ratio of DNA to cytoplasm affected the biophysical properties within the cell, and it was associated with a phenotype such as senescence. So for this study, we wanted to know how the biophysical properties of the cytoplasm, such as how diluted or how crowded these cells are, change with cell size. We measured intracellular diffusion to probe the crowding of the cytoplasm and asked, how does diffusion change in cells that are not only overly large but overly small? And further, how would intracellular diffusion change if we could also alter this ratio of DNA to cytoplasm in these larger cells by also probing DNA content in these cells? To measure intracellular diffusion, we are using GEMs, which are genetically encoded multimeric nanoparticles, as my colleague Arthur introduced this morning. They were developed in the Holt lab at NYU, and what they are is that they're engineered from protein cages that are found in non-eukaryotic organisms. These protein cages are assembled from encapsulin protein monomer that's fused with GFP, and this self-assembles into a 40 nanometer particle. And they are in the size range of abundant protein complexes such as the ribosomes. And so we have these GEMs in fission yeast, because we've chosen to use fission yeast as a system to ask these questions. Fission yeast is genetically retractable, they have simple cell geometry, and we know a lot about how they grow and divide. On the left, there is a picture of them there. In the middle is a movie of pombe with GEMs inside of them. And some things that you should know about fission yeast is that they're rod-shaped, and the way they grow is along the long axis. And they grow until they reach a stereotypical length and divide, and we exploit this cell cycle to alter cell size and the DNA to cytoplasm ratio. So first, we altered cell size using mutants that shorten or extend the G2 phase. So this WE1 cell here, this WE1 mutant here divides at a shorter length, while the CUC25 mutant divides at a longer length. And you can see that these cells have different sizes but the same DNA content. Thus, we have altered this ratio of DNA to cytoplasm. Here are the different types of cells. This is the maximum projection of our GEMs over time. We use single particle tracking of these GEMs to derive an effective diffusion coefficient. And then we see here across the experiments, GEMs in small cells are slower than the wild type, and GEMs in these bigger cells are going faster than wild type. And then when we break this down to the single cell level and plot cell length, which is our measure of size, and their corresponding diffusion, we observe a general positive relationship between length and diffusion. So now that we probe cell size, we want to interrogate this diagonal here by preserving the DNA to cytoplasm ratio of our wild type cell, but to do so in bigger cells. The way we did this is we looked at cytokinesis mutants, the cytokinesis mutant SID2. So the cells do not divide, but they still go through the other stages of the cell cycle, so they get longer and they also get more copies of DNA. And here they are here. We have our control cell, and we inhibit cytokinesis with induction of a drug. The longer that this drug is in there, the cells do get longer and they get more DNA. This is the nuclear envelope in red. And here I'm just showing you on the population level, indeed, cells do get longer and they also do get more nuclei. And when we look at the GEMS diffusions across these cells, we see that even though the cells are getting larger, they don't have a change in GEMS diffusion compared to our control. So overall, what I've shared with you today is that we have a system to alter cell size and the ratio of DNA to cytoplasm, and to measure intracellular diffusion in these cells. We believe our results suggest that the DNA to cytoplasm ratio determines the biophysical properties of the cytoplasm, and that we observe in an overly large cell, like our CUC25 mutant, with a lower DNA to cytoplasm ratio, this suggests our cytoplasm may be diluted, and by converse, when we have our overly small cell, like our WEE1 mutant, we have a higher DNA to cytoplasm ratio, and indication that this may have a more crowded cytoplasm. But if you preserve this ratio in our larger cells, we see that the intracellular diffusion here doesn't change as seen here with our cytokinesis mutants. Looking forward, we are most excited to be working with the Jan Skotheim lab at Stanford to ask how the proteome is affected by cell size and this ratio of DNA to cytoplasm. Together with Matthew Swaffer and Michael Lands, we're looking particularly in our cell size mutants to ask which proteins have changed in concentration and what these protein changes can tell us about the physiological state of the cells, and if we see phenotypes such as senescence. So finally, I'd like to acknowledge the Fred Chang Lab, every single one of them, and my thesis committee for all their continued support and advice over the years for this project, as well as the Skotheim Lab for their work with us on characterizing proteins in fission yeast, and the Holt Lab for their guidance with working with GEMS. And thank you all for listening.